Hello, my name's Dr. Peter Walley and I'm a research scientist at Warwick Crop Centre. My primary interests are vegetable crop improvement, and I tend to concentrate on brassicas, lettuce, carrot and onion. I work with the Vegetable Crop Improvement Network, and we aim to develop and address traits of importance to industry, and much of our work is industry-led. Behind me you can see a field of carrots. These are some of the carrot diversity set, and we also develop genetic resources and seed resources that are then used in whether it development of other projects or can be taken up by industry. So here we are in the Genetic Resource Unit based at Warwick Crop Centre, and here we keep the majority of our seed collections under standard conditions for long-term storage. Once a particular target trait has been identified and the assays developed to phenotype the target crops, we then need to consider the genetic variation that we wish to capture in order to improve the crop and improve the phenotype. And within the vegging project, we have developed fixed populations that capture genetic variation from wild species, which allows to propagate them indefinitely and to replicate them in trials. We also used fixed mapping populations, biparental populations, in order to do further genetic analyses and for QTL identification and line development. So here we are in the lab, and it's in the lab where we do DNA extractions for genotyping. Once we've measured the desirable phenotypic variation within our different plant lines, whether it be mapping populations or natural diversity sets, one of the things that we need to do is to link that phenotypic variation with genetic variation, and that involves taking DNA samples from the lines, which we do in the lab, and using the DNA for genotyping. Once we've got the genotyping data, we then use the genotyping data in conjunction with either genetic maps, or we make genetic maps from the biparental populations, and then we map the phenotypic variation onto the representation of the genome to look for the areas that control the target trait. These areas then, known as quantitative trait loci or intervals, are then used with the linked genetic markers to select progeny within the population of advanced lines. So once we've developed assays and phenotyped the desirable plants, whether it be mapping population or diversity set, we then have to develop the lines further, and this usually involves either developing double haploid lines from crosses F1s, or for the diversity fixed foundation sets in brassica, we took the wild species, crossed it to a recurrent parent, which is a standard line for all the crosses, and then made double haploid lines from those. After seed regeneration, these lines are then fixed as immortal lines, capturing the wild species genetic variation. And here you can see we've got some of the brassica lines that we're collecting more seeds from, and other lines that are advanced lines from populations that we are taking through further generations to fine map traits of interest. We've got some lettuce lines in here, and these are for some of the insect work that we're doing to capture variation from wild species of lettuce with domesticated lettuce to look for insect resistance.